Hello, everyone. Welcome to the Sun Devils study of science and society, where a group of Arizona State University students introduce, investigate, and have a friendly discussion about controversial hot science topics of the present and future. Whether you just found this podcast today or have been following us along our journey since the beginning, I just want to say thank you for supporting us and allowing us to share our passion of science with you guys. In today's episode, we will be dissecting the big deal behind designer babies. I'm Felicity, and I'm here with Preeti, Abigail, Noel, and Buthiana, who will guide us through the scientific process in terms of the CRISPR mechanism and also help us evaluate the ethical discourse surrounding this technology. To begin, let's look at a hooked text video introducing today's topic. So, Buthiana, could you tell us a little something about the science behind these CRISPR babies?
Yeah, of course. So, CRISPR, also known as clustered regularly interspaced short palindromic repeats, was discovered in E. coli in 1987 by Yoshizumi Ishino. So, the function of these repeats was not determined at the time due to insufficient DNA sequencing data. But in 1993, Francisco Mojica became the first researcher to name the CRISPR locus what it is now referred to by. Mojica also predicted that CRISPR was an adaptive immunity response. This was confirmed by Mojica and his coworkers when they found that after a virus infects a prokaryote, spacer sequences in CRISPR are transcribed, and the resulting product guides the Cas protein to cleave DNA or RNA viral sequences. This role of Cas proteins was discovered by Makarova and his colleagues, who suggested that the function of the CRISPR-Cas system is comparable to that of RNA interference, where a complex of proteins cleaves mRNA, thereby silencing certain genes. They also found that there are different types of Cas proteins, such as Cas9, which only cleaves DNA, and Cas13, which only cleaves RNA. A critical part of the CRISPR-Cas system are protospacer adjacent motifs, also known as PAMs. PAMs are short sequences found adjacent to a sequence being targeted by the Cas protein. In this way, PAMs act as recognition signals for the Cas protein when cleaving DNA. In 2012, the CRISPR-Cas9 complex emerged as a genome editing tool. The CRISPR-Cas9 system has two important components, the DNA cutting protein Cas9 and an RNA molecule known as guide RNA. First, the Cas9 complex locates and binds to the PAM sequences. The guide RNA then proceeds to unwind a specific section of the DNA double helix and also locate the target DNA sequence to be cleaved. The Cas9 protein can then cut the DNA by inducing a nick that causes a double strand break. At this point, the cell might try to fix this break. However, this process is extremely prone to error and introduces mutations that can knock out an entire gene. According to Medline Plus, this system allows for genetic material to be added, removed, or altered at particular locations.
Oh my, that's a pretty hefty quote. Noel, do you have any say in that?
So the quote in here states others, such as Sandel, argue against it in the basis that it indicates that parents have an undesirable attitude that are at odds with the norm of unconditional love for one's child. And that's basically just saying parents are more attached to their kids who carry more phenotypic attributes that match their own than their kids who do not. One popular person who plays into this is Kim Kardashian. When her and Kanye were thinking about having their last kid, they were rumored to have genetically engineered the embryo to make the baby more attractive and in line with social norms in order to have the most beautiful child in comparison to her sisters. This was just a rumor, however, but it shows in what lengths society can go to just to have a child look more like them or more in line with social norms than what is normally up to in genetics.
So, Preeti, what are the ethical dilemmas behind this hot new tech?
Yeah, so there are a few concerns about the ethical dilemmas of this technology, and an article from The Guardian discusses how IVF treatments have provided insight into what could possibly happen with the genetic editing of embryos, as it could quickly lead to negative consequences. This also brings up the possible consumerism of genetic editing, as the focus could shift from helping couples looking to expand their families or have healthy children to attracting customers. This relates into how some researchers are worried that the use of the procedure will change from improving health to non-therapeutic enhancement, such as designing the perfect baby with characteristics and visuals that are more aesthetic to the parents. Further concerns with CRISPR come from the fact that it can't be guaranteed that undesirable effects won't arise post genetic editing and the bioethical issue of informed consent, as the babies affected cannot consent to the procedure and have no say in their future if undesirable outcomes are obtained. Furthermore, there is a socioeconomic barrier to these procedures, as genome editing is very expensive, making the technology more accessible to the wealthy individuals and preventing families without such wealth to have access to this life-saving tech. So while there are many benefits to the technology of genetic editing, we can see that there are also ethical dilemmas that must be considered before genetic editing is approved in embryos.
just to go over the CRISPR technology, it was first used on gene-edited babies for HIV resistance, and this was researched by the Chinese researcher He Jiankui. It was said that the scientific community actually did not have much of a supportive reaction, and as of now, his research has yet to be peer edited as well as published.
Well, that's all the time we have for today, folks. So as always, thank you for tuning in to Sun Devil's Study of Science and Society. Let us know what you think on Twitter with the hashtag Sun Devil Study SNS, and we'll see you next time. Bye.